Hello everyone, today we're going to be talking about the kidneys. We're going to start off with the purpose of the kidneys, which is to regulate fluid levels, regulate ion levels in the blood, and to filter waste out of the blood. So when we look at each kidney is the size of a closed fist. They're located just above the small of your back between the T12 and L3 vertebrae, with the bottom of the kidney being right about where the bottom of your rib cage is. They're located towards your back, so if you looked at a cross-section of your body, you'd be able to see that they're located more towards your back since the kidneys are behind all the major organs found in the abdominal cavity. Located on top of each kidney are your adrenal glands. These release adrenaline. The adrenal cortex also regulates renal function by producing aldosterone, which triggers sodium reabsorption. And below and out of the kidneys, you'll find a ureter exiting each kidney that leads to the bladder and out of the body through the urethra. The kidneys are well protected by layers of fat, muscle, and ribs. The fibrous capsule directly covers each kidney and has 3 layers. The fibrous capsule, also known as the renal capsule, which is a dense layer of irregular connective tissue. On top of that is the renal fat pad, which is made up of adipose tissue. And on top of that is the renal fascia, which is a tough anchoring layer that firmly anchors the kidneys to the posterior abdominal wall in a retroperitoneal position and holds them in place. When we look at the kidneys, we'll see 3 major parts. The cortex, which is the outermost layer and is also shaped as a C, so easy to remember, the cortex C. The medulla layer, which is the middle layer, so think medulla, middle layer. And the renal pelvis, which you can remember by thinking it leads to the pelvis through the ureter, then the bladder, and out through the urethra. By the renal pelvis, you'll also find the renal pedicle, which is where you find the arteries that supply the kidneys with blood, and the vein that takes blood out of the kidneys, and the renal pelvis, which takes waste to the bladder and out of the body. The renal cortex is a thin layer, but it's filled with nephrons. Each nephron is a functional unit of the kidney. This just means that the nephron is where the blood is actually filtered. Each kidney has about 1 million nephrons, so if you multiply the filtration capacity of each one, you get the filtration your body needs to stay regulated and free of waste. A lot of blood goes to the kidneys. About 20% of the cardiac output goes to the kidneys. So how does the blood get in and out? The medulla is where this is made possible. The renal artery brings in blood and runs through the renal column and into the renal cortex, where then arterioles lead to the nephron, and then the venules back out to the renal veins and out to your body. The filtrate goes on through these triangle-shaped tissues called the renal pyramids. From there, the filtrate goes through many collecting ducts that get larger and larger until they flow out through the renal pelvis and onto the ureter and the bladder and the urethra. When the blood enters the kidney, it enters via the renal artery, which splits off into smaller and smaller arteries called arterioles, which enter a nephron through the efferent venule, split off into capillaries, and here they bundle up into special type of capillaries called glomerules, which you can remember by thinking of glob. So glob, glomerules. This glob of blood vessels, they're super tiny. We have about 2,000 glomerules per square inch of kidney. The capillaries here have tiny holes. They're known as fenestrated capillaries, and they let smaller particles like glucose, amino acids, and water through. But they're too small, so they keep red blood cells, immune cells, and other big proteins from leaving. The capillaries here tangle up in the glomerules and are encapsulated by Bowman's capsule, which is several layers of epithelial cells, which pretty much make sure that waste products go down the right tubes. This group here is known as the renal corpuscle. Here we see the first steps of the urine formation, which is filtration. And that's the initial movement of molecules from the glomerular capillaries into Bowman's capsule. Urine production starts with the filtration of blood by the glomerular capillaries. The volume of filtrate formed by both kidneys per minute is known as the glomerular filtration rate, or the GFR. On average, 160 liters a day of filtrate is made, but because of reabsorption, only about 1 to 2 liters of urine are actually produced per day and excreted. GFR is based on 3 different pressures, and those would be the hydrostatic pressure of the capillaries, HPC, colloid osmotic pressure of the capillaries, OPC, and hydrostatic pressure of the interstitial fluid in Bowman's capsule, or the HPIF. Net fluid movement will be in direction of the lower pressure, where, on the contrary, colloid osmotic pressure is the pressure that pulls water towards it because of the higher solute concentrated. So if we want to figure out what that GFR is, and see what direction the water is going to move in, we need to calculate the net filtration pressure. So to do so, we're going to subtract the inward driving forces from the outward driving forces. Glomerular filtration occurs because glomerular hydrostatic pressure exceeds the hydrostatic pressure of the interstitial fluid in the Bowman's capsule, along with the glomerular colloid osmotic pressure. Branching off Bowman's capsule is a long tube that starts in the renal cortex and winds around and dips down into the medulla, into the loop of Henle, comes back to the cortex where it empties waste products into a collection duct. The closest part of the corpuscle is the proximal tubule, while the farthest is the distal convoluted tubule. The middle section is the loop of Henle. This gets its name because it crosses from the cortex down through the medulla. After the filtrate makes its way through a collection duct that takes the filtrate to the renal pelvis. If we go back to the glomerulus, the capillaries are jumbled up inside Bowman's capsule, but they still need to exit. The same capillaries go to the efferent venules, wind around the different tubules known as peritubular capillaries, and then go by the loop of Henle as the vasa recta, and later on become the renal vein. This hectic route is necessary because a lot is dumped into Bowman's capsule, including things that we want to keep in our body, like water and glucose. Right after Bowman's capsule, we see the second step of urine formation, which is reabsorption. And that's the movement of molecules in the renal tubule back into the capillary, or the bloodstream. So this filtered fluid collected by Bowman's capsule enters into the PCT, which is the proximal convoluted tubule. The PCT is called convoluted because it's kind of a tortuous path. It's convoluted. The lumen of the PCT is lined with thousands of hair-like structures that increase the surface area to maximize the absorption and secretion of solutes like sodium, calcium, and glucose. The most essential function of this portion of the nephron from here is that filtrate enters the U-shaped structure, which is referred to as the loop of Henle. The descending and ascending portions of the loop of Henle, also called the nephron loop, are just continuations of that same tubule. And they run adjacent and parallel to each other. After making that little U-turn, in the loop of Henle, water flows out in the descending portion of the loop, so water out through the descending portion, but in the ascending portion, no water flows out. Instead, ions flow out. Sodium, potassium, calcium, magnesium, and chloride flow out. Because the ascending portion dumps out all these ions, it makes the descending portion of the loop, which is right next to it, more salty. Since water follows salt, it causes water to flow out of the descending portion, but not the ascending portion. This happens because of osmolality, which is the measure of the amount of solutes dissolved in a liquid. Water flows from low osmolality to high osmolality. In the loop of Henle, this works by using the passive concentration gradient to move water by having it follow the small molecules of ions across barriers. From here, we move on to the DCT, and we see the third step of urine formation, which is secretion, and that's the movement of molecules from the capillary into the renal tubule after filtration. The DCT, or distal convoluted tubule, just like the PCT, is very tortuous, it's very convoluted, but it's shorter than the PCT. Its lumen is also lined with hair-like structures, and it connects with the collecting duct. The collecting ducts are continuous with the nephron, but not technically part of it. In fact, each duct collects filtrate from several nephrons for final modification. Collecting ducts merge as they descend deeper into the medulla and drain into a papilla. Collecting ducts are lined with membrane protein channels called aquaporins. And just like their name suggests, with hormonal regulation, these channels allow water to pass from the duct lumen through the cells and into the interstitial spaces. This process allows for the recovery of large amounts of water from the filtrate back into the blood. From here, the filtrate moves down the renal pyramids and into the renal pelvis and down the ureter, where we can see the final step of urine formation just after reaching the bladder and exiting through the urethra, which is excretion. Thank you again. I know the kidneys are not the easiest thing, but you can rewatch this video, you can slow it down, and just look at the different steps. Thanks again.